Hello, good day everyone. Hope you feel good. Are you ready to start the lesson? Is the sound clear? Okay. The general traits of the fungi are gone. Homotheles and heterotheles in fungi. Based on the compatibility in sexual reproduction, the fungal hypha distinguished into two types, homothelic and heterothelic. In homothelic fungi, fusion occurs genetically similar strain for mating types. If the two strains of fungi is similar in genetic, they can fuse and reproduction. In such forms, meiosis results in formation of genetically identical spores. In the heterothelic fungi, fusion occurs between the genetically different mating type or strain. One strain called and another strain called mice. In heterothelic fungi, fusion occurs between the genetically different mating type or strain. The strains are genetically compatible and distinguished as plus and minus strain. Okay, I refresh the sound and come back. Sound is okay? Is the sound clear? Miss Marve, please refresh your system. But in homothelic fungi, fusion occurs between the genetically similar strain and or mating type, similar mating type. In such forms, meiosis result in the formation of both strains in equal numbers. Importance of fungi. Fungi are agents of biodegradation and biodeterioration. Saprophytic fungi. The fungi that alive on non-organic material and dead material. Saprophytic fungi utilize dead organic materials as source of nutrients and are responsible for the biodegradation of organic material in our environment, particularly plant material in the form of leaf litter and other plant debris. These fungi survive on dead organic material. Sound bad for all of you? Okay. This okay? Is the sound better? Is the sound better? Is the sound okay? Is the sound got better? Okay. Saprophytic fungi utilize dead organic material as source of nutrients and are responsible for the biodegradation of organic material in our environment, particularly plant debris, plant material in the form of leaf litter, and wood or cellulose, lignin, chitin, and the other. Such fungi play a vital role in recycling essential elements, particularly carbon. Fungi are very effective and efficient biodegraders because of the wide range of extracellular enzymes they produce. The hyphae of fungi produce extracellular enzymes that release in environment. This enzyme capable of degrading complex polymer such as cellulose, proteins, and lignin. Fungi are responsible for the majority of plant disease and several diseases of animals, including humans. Some fungi are actively parasites in humans and other animals, while others induce severe allergic reaction if their spores are inhaled, resulting in attacks of asthma of fever. Some fungi, such as Aspergillus, the spore of Aspergillus, produce allergy in humans. On the other hand, Phytophthora infestans is the causal agent of late blight disease in potatoes. This is an important disease in potato, and this disease reached epidemic proportion across Europe in the middle 19th century and result in the Irish potato famine. Fungi used in industrial fermentation process. Yeast and mycelial fungi used in a variety of industrial fermentation process. For example, Saccharomyces cerevisiae. Saccharomyces species used extensively in brewing beers and wines, as well as in bread making and in produce cheese and food industrial. Fungi also used in the commercial production of many biochemicals. Aspergillus niger used in the large-scale commercial production of citric acid. Many fungi provide us with a direct source of food. Some yeast and mycelial fungi cultured on a large scale and then undergo further processing to provide various protein-rich food products for human or livestock. For example, mycoprotein produced commercially from the mycelial fungus Fusarium venenatum. The mycelium harvested and processed to provide a protein-rich meat substitute in a range of convenience food. Some species are cultivated for their edible fruiting bodies. You know, agaricus. One of mycelial fungus that do you have my sound? Okay. Agaricus is one of the mushrooms used for food. Fungi used in bioremediation. Some species of yeast and mycelial fungi are used in processes aimed at reducing the concentration and toxicities of waste materials, particularly from industrial processes, before those wastes are released into the environment. A process known as bioremediation. For example, Aspergillus niger is used to break down tannins in ternary influence to less toxic compounds. Some fungi prove highly beneficial in agriculture, horticulture, and forestry. For example, some species form symbiotic relationships with the roots of plants known as mycorrhiza. There are the roots of trees and some fungi that called mycorrhiza grows with these roots in tree, and absorb the water and mineral from the soil, and these mycorrhiza get food from the tree. Other fungal species also used in the biological control of insect and nematode pests, weeds, and pathogenic microorganisms. For example, the fungus Bavaria bassiana used to control a number of insect pests. Some fungi prove highly uh, beneficial in cheese manufacture. Cheeses such as uh, Gorgonzola, Roquefort, Stilton, and blue cheese have fungal colonies that give this cheese their distinctive flavor. Some fungi prove highly beneficial in medicine products. Penicillin, perhaps the most famous of all antibiotic drugs, and derived from a common fungus called Penicillium. Many other fungi also produce antibiotic substances, which are now widely used to control disease in human and animal population. The discovery of antibiotic uh, revolution revolutionized health care worldwide. Fungi cause food spoilage. Already, it has noted that fungi play a major role in recycling organic material. The fungi, which make our bread and jam go moldy, are only recycling organic matter. Even though in this case we would prefer that it did not happen. Fungal damage can be responsible for large losses of stored food, particularly food that contain any moisture. Diversity and classification of fungi. Fungi grow in a wide range of habitats, such as extreme environment, including deserts or areas with high osmotic condition or sugar and salt concentration, ionizing radiation, as well as in deep sea sediments. Some of fungi can survive the intense UV and cosmic radiation in country during space travel. Must grow in terrestrial habits, though many species grow partly or solely in aquatic habits, such as the chytrid fungus, a parasite that has been responsible for a worldwide decline in amphibian population that called Batra cocyridium dendrobotidis. This fungus spent part of its life cycle as a motile zoospore, uh, enabling it to propel itself through aquatic habits and enter its amphibian host. Other examples of aquatic fungi include those living in hydrothermal area of an ocean. Therefore, fungi have a worldwide distribution. Fungal species are historically being distinguished by many methods and concepts. Classification based on morphological characteristics such as the shape and size of spores, 23 or fruiting structure has traditionally dominant fungal classification. Species may also distinguish by their biochemical and physiological characteristics, such as their ability to metabolize certain biochemical material or their reaction to chemical tests. Recently, the application of molecular tools, including DNA sequencing and phylogenetic analysis to study diversity, has greatly improved the resolution and added robustness to estimates of genetic diversity within various taxonomic groups. In addition, fungal taxonomy depends on microscopically and microscopically observable characteristics. Macroscopically, traits such as color and shape of colony. Microscopically, traits such as the shape of spores and other morphological structure of fungi. The phenotypic approach, such as color of the growth, rate of growth, method of spore production, and high fall septation, all aid in assigning a fungus a name. The colony of fungi on growth media have a color. That this color used for classification of fungi are the rate of growth in a day, center of colony, the fungi grows, and determine this growth in a day. More technologies that are newer have aided in both identifying new organisms and recognizing the various forms of the same organism. Electron microscopy allows recognition of a structure not visible by a light microscopy. Physiological and biochemical techniques are being applied as well as identification of secondary metabolites such as enzyme, toxin, polymerase chain reaction or PCR allows comparison of the DNA or RNA structure of organisms, including fungi. In the earliest taxonomy, there were only two recognized kingdoms, including plants and animals. This two-kingdom system was used until Whitaker in 1969 proposed that organisms be classified into five kingdoms, including Monera or bacteria, Protista, mostly alga and protozoa, Planta or plants, uh, sound is bad, sound is not clear. Okay. Is the sound got better? The internet is getting worse every day. The internet is too slow in Iran. Whitaker distinguish five kingdoms. There are including Monera or bacteria, Protista, mostly alga and protozoa, Planta or plants, Myceta or fungi, and Animalia or animals. Fungi belong to domain Eukarya and kingdom Myceta. The classification of fungi as proposed by Ainsworth in 1973 commonly follow. There are more than 60,000 fungal species known. Classification of fungi is by their method of both sexual and sexual reproduction. It seems likely that fungi are not a monophyletic group. Historically, they have divided into four taxonomic divisions: Zygomycota, Ascomycota, Idiomycota, and Deuteromycota. The last group, Deuteromycota, is not a monophyletic group and thus has no standing in a modern classification scheme. The modern systematic grouping, including the Zygomycota, Ascomycota, and Basidiomycota, as well as the Chytridiomycota. Schroer in 1893 classified the fungi into four classes: Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes, Sacchardo in 1931 classified the fungi into six classes: Schizomycetes, Myxomycetes, Ascomycetes, Deuteromycetes, Basidiomycetes, and Phycomycetes. Bessey in 1950 classified the fungi into three classes: Schizomycetes, Myxomycetes, and Eumycetes that you see in this chart. No, not necessary. Classification, yes. Classification is important. Only you just have to keep the last category. You don't have to memorize them all. Mycologists have traditionally studied these types of organisms and divided based upon phylogenetic relationship, and that these relationships are kingdom fungi or true fungi, kingdom Straminiplea that called water mold, kingdom Mycetozoa that called slime molds. Only memorize this slide. Okay, only memorize this classification. Kingdom fungi or mycota. Phylum Chytridiomycota, Zygomycota, Gelomyromycota, Ascomycota, Basidiomycota. Form phylum Deuteromycota or fungi in perfecti or imperfect fungi. This slide is important and you must memorize it. Kingdom Straminophila or Chromista. Phylum Omycota and two Hyphochytridiomycota and Labyrinthomycota. Kingdom Mycetozoa. Phylum Myxomycota. Dictyo Steliomycota, Acrasiomycota, Plasmodiophore mycota. Chytridiomycota. This group or these fungi produce zoosporic flagellate cells during their life cycle. They have flagellum. Two classes exist. Class A, Chytridiomycetes. Class B, Plasmodiophoromycetes. You must memorize these four taxon: Zygomycota, Ascomycota, Basidiomycota, and Deuteromycota. This slide, not important. Not important. This slide is important and you must memorize it. Okay, I refreshed. Is the sound got better? Okay. You must memorize this slide and this slide. Okay? General characteristic of Chytridiomycete or Chytridiomycete. The Chytridiomycete or chytrid are usually aquatic, either marine or freshwater, and they survive in aquatic condition. The chytrid are mostly single cult forms. Chytrid have no light sensitive pigments. Presence of posteriorly uniflagellate called zoospore. The flagella are of flash type. They have a single whiplash flagellate. Whiplash flagellate. Fungi. Chytrid observe nutrient from environment. Chytrid have cell walls composed of chitin. Sexual reproduction is by isogamy to oogamy conjugation. Asexual reproduction by uninucleate zoospores. Chytridiomycete is classified into the following important order, and there are chytridiales and blastocladiales. Thallus and life story diversity in Chytridiomycete. Unicellular holocarpic may produce rhizoid that mainly serve to enter thallus rhizoid-like nuclei. This is a chytrid that usually is unicellular, holocarpic, and may produce rhizoid. They look like roots of plants. filamentous eucarpic cenocytic mycelium. Cenocytic mycelium not divided. Aseptate hypha. If the hypha have Okay. Flagellate stage of life cycle. Zoospores, asexual reproduction. Planogametes, sexual reproduction. And both zoospore and planogametes possess a single posterior whiplash flagellum. In chytrid, zoospores and planogametes possess a single posterior uniflagellate. Yes. Zoospore containing body is a spherical or pear-shaped sac. Called zoosporangium. Sexual reproduction has not observed in a majority of the chytrids. A thick-walled resting spore or resting sporangium formed after sexual reproduction. Chytridials classified into many important families, from which Syngetriaceae, for example, Syngetrium endobuticum. Yes, of course. Okay, sure. Chytridials found in aquatic and they live in water condition or moisture condition. Some of them parasitically on algal vesicular plants such as Syncytium endobioticum that the fungi parasite on potato. Many forms are endobiotic. The endobiotic chytrid live completely within the cell of the host. Endobiotic chytrid live completely within the cell of the host. The epibiotic chytrid live on the host surface. Asexual reproduction takes place with the help of zoospore, which are posteriorly uniflagellate. Zoospore containing body is spherical or pear-shaped, called zoosporangium, and the zoospore exists in the zoosporangium. I refreshed it. Okay. Is the sound got better? Okay. It is the end of Ramadan and the time of Iftar is near. Please pray for me. Lesson are enough for today because the lesson was difficult, and I think lessons are enough for today. Okay. Happy Eid al-Fitr. I send a lecture in WhatsApp group after the class. عید فطر شما هم مبارک. Thank you, Miss Adnan. Thank you, Mr. Qasim. Thank you, Miss Marve. Thank you all.